Hello, my name is Mark Sorrells. I'm a small grains breeder at Cornell University, and today I'm going to talk about some of the work that we've been doing using molecular breeding and high-throughput phenotyping. Crop improvement began with the hunter-gatherers who practiced mass selection and domesticated the species that became our crops today. Clearly, they were successful with no knowledge of the underlying genetics and that were controlling the traits that they were selecting. Over time, plant breeding has used new breeding methods to increase the heritabilities of the various economic traits. Today, the focus is on the use of molecular markers that improve heritability and allow us to improve our prediction accuracy. In fact, plant breeding is a predictive science, and novel breeding strategies are driven by technology and new knowledge. Complementary to marker-assisted selection is genomic selection. Curiously, this breeding method does not rely on an understanding of the underlying genetic control of the traits we're selecting. So over time, breeders have not abandoned old breeding methods, but instead they have adapted them to new technologies and integrated them into new strategies. One example that I'm going to tell you a little bit about is the adoption of aerial and ground-based imaging systems to generate secondary traits that are correlated with biomass and grain yield. Plant breeders are facing a number of challenges. In particular, we are realizing a yield plateau in many of the crops in various regions. Connecting phenotype to genotype is more important than ever now that we have good marker systems. And the limitation now is generating enough quality phenotype data to be able to improve our prediction accuracies. We want to be able to understand and predict genotype by environment interaction. We need to predict phenotypes under increasingly extreme weather conditions brought on by climate change. And we need methods of phenotyping that generate information on a large scale and cheaply comparable to DNA sequencing with reduced cost. But if we're going to meet these challenges, we need to embrace new technologies. First, I'm going to tell you a little bit about a project that was carried out by Jessica Rakowski, where she used genomic selection and conventional phenotypic selection to improve adult plant stem rust resistance in wheat. And this was for the strain called UG99. She carried out phenotypic selections in the Kenya stem rust nursery for one season, and then compared that to 2 cycles of genomic selection carried out in the lab here at Cornell University in the absence of the pathogen. In her experiment, 1 cycle of phenotypic selection was equal to the 2 cycles of genomic selection verified in 2 replicates. But the advantage to the genomic selection is that she didn't have to go to Kenya and score large nurseries for this dangerous pathogen. Instead, it was carried out in the lab in a much shorter period of time. Accompanying the gain from selection was an increase in inbreeding. And of course, this is a disadvantage because when you reduce the genetic variation, you reduce the opportunities for increased genetic gain over time. We addressed that limitation by using a method called genomic selection with optimum contribution selection. This method is used to reduce the inbreeding, and it was set up so that we could reduce the inbreeding by 50% in each generation. This approach was successful. As you can see in this slide, we actually had more genetic gain from the constrained selection shown in the red line than in the conventional genomic selection shown in the green line. Simultaneously, we were able to reduce the rate of inbreeding over selection cycles. Here again, reduced inbreeding was realized with a constrained selection. The second project that I want to tell you about was carried out by Dr. Margaret Krauss, and this was a collaboration with CIMMYC to use hyperspectral imaging to produce phenotypes that are used as markers in the genomic selection framework. And this was used to then try to improve the prediction accuracy for grain yield in wheat. In this experiment, we gathered reflectance values for 62 different wavelengths, and each of these lines is the correlation between those wavelengths and grain yield for different dates in the optimal environment. Heritabilities for this project were quite good, ranging from about 0.4 to 0.6 for both grain yield and the spectra. An example for one environment is shown here, where in the orange you see the prediction accuracy for markers only. In the green, the prediction accuracy for only the spectra. And then the blue shows the prediction accuracy when we combine both the markers and the spectra. So you can see that over the season, at different time points, the prediction accuracy increased, and until in the last two columns, you see all time points put together gave us the best prediction accuracy overall. When she had analyzed the data across environments and compared the prediction accuracy for markers only, pedigree only, markers plus the hyperspectral reflectance, or the pedigrees plus the hyperspectral reflectance, she found that the prediction accuracy using the markers plus the hyperspectral reflectance gave the best or equal to or the best prediction accuracy over all environments. So I'd like to stop there and just acknowledge a number of important sources of support, including the Bill and Melinda Gates Foundation, the USDA, and my Cornell Small Grains breeding team at Cornell University.